This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 314, recorded on June 27th, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from northern Michigan, Michele Swanson.
Hello, great to be with you all, and in this lovely sunny weather where it's about 70 degrees.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone, and today we have a very swamp-like weather of 92 degrees, and it's when you walk outside, you're walking in air you can feel. It's that humid.
And from St. Louis, Missouri, Petra Levin.
It's great to be here. We have quite the humidity, but we finally cooled off. It was over 100 yesterday, I think, at some points, or the day before yesterday. And yesterday it rained all day, which we really desperately needed. The plants you can tell they're happy now outside. And even though it's really humid, it's only 88 right now. So, yeah. Well, for Missouri in summer, 88's like fine
cool
weather.
Here in New Jersey, it is 29°C. We had this incredible storm rip through. I was doing a live stream last night, and the power cut out momentarily, so I had to reboot everything. But all these trees are down all over the place. Really crazy. I learned on my travels that there are only 3 countries in the world that use Fahrenheit.
Oh, gosh. Are we that backward?
The U.S. and 2 others. I forgot which they are. But I saw this graphic on Instagram, countries that use Fahrenheit, and there were just 3. Everybody else uses centigrade. You know, we tried to switch, but it didn't work in the U.S.
I feel like we should be able to switch temperature. Distance is very difficult. Like even England hasn't, I think, switched their distance and speed for road travel, but I feel like we could at least add centigrade. So.
Well, that makes sense. Look, as scientists, we use metric all the time, right?
So those countries, to save people from Googling, are the Bahamas, the Cayman Islands, Liberia, Palau, the Federated States of Micronesia, and the Marshall Islands, which is a territory of the U.S.
It's like 6 different places, and they're all pretty small. So majority, Europe, Asia, Africa, Australia, they're all using Celsius.
They told us in elementary school that we were going to need to use metric, and they taught us metric.
Yeah.
But I don't know if they taught my kids that. I don't think they did in elementary school.
Well, and the weather reports that we get certainly don't give us degrees Celsius.
No. And the other thing they taught us, though, was different bases, like base 12, base 10, in addition to base 10.
And base 8.
Base 8. In addition, and they just barely taught us binary. And they said in case aliens come and they have 12 fingers. So nice. I guess I learned both centigrade and potential for aliens with 12 fingers who count in base 12 at the same time. So I know centigrade better, though. I don't think I could count in base 12.
So you're preparing to take us to your paper today by talking about alien life.
Yeah.
Exactly. I'm prepared to talk about alien life very briefly, actually. So today I have a snippet, which is really it's from the New York Times, and it's an excerpt from a book by Faris Jabbar. And he's a contributing writer, and he's an author of a new book called Becoming Earth, How Our Planet Came to Life. And it's really thinking differently about the Earth as we know it, because in my mind, probably from what I was taught, I think of early Earth and as being kind of similar, at least on the ground, like there was ground to stand on, and the ground was kind of like the ground it is now. Maybe no soil, but like rocky. And really, it's our atmosphere that's been transformed by life, right? Oxygen coming from cyanobacteria and things like this. But this book really makes the case, and as does this article in the New York Times, which we'll link to, is that our Earth has really been transformed, and the way even what's underneath us in the crust, in this article is the focus of the crust, has been transformed by microbial life. And
there's
so
a lot going on under our feet.
There's a lot going on under our feet. And the article actually starts off really in an exciting way because he descends into a mine in Lead, South Dakota, the Homestead Mine, which has tunnels that are up to 370 miles in length. But most importantly, they go 1.5 miles below the surface. And it's the deepest and most productive gold mine, or was the most productive gold mine in North America. And it's now the property of the US government. And it's mostly used by physicists. And I forgot this about physicists, but some who are doing certain kinds of measurements, they need to be shielded from cosmic rays. So if you've ever taken a Geiger counter, or when I learned about the Geiger counter, right, you turn on a Geiger counter, you hear it clicking, and that's just sort of transient radiation coming from our atmosphere, actually from space, which is a good reminder. So to get away from that and other kinds of forms of cosmic energy, physicists really need to go very deep. But more recently, it's been used by biologists to find organisms that transfigure rock minerals into other things just through their metabolism. And this descent into this mine takes about 10 minutes. It's I think it's about a mile down. It takes 10 minutes. And he goes actually with a graduate student named Caitlin Cesar, who's a graduate student at Northwestern in the lab of Magdalena Osborne, who's in Earth and Planetary Science. And they really study these microbes that transform the Earth. So he has this guide, Caitlin Cesar, and the first thing that they see is a spigot, a plastic spigot, right? You go 10 minutes down in an elevator and you see a plastic spigot and there are white fibers coming out. And these are really, in my mind, I'm like thinking of these giant bacteria, but they're actually fibers made from, the author compares them to strings from poached egg, and they're from a group of bacteria called thiotrix, which are sulfur oxidizing gamma proteobacteria. So they're in the same big group as E. coli or Pseudomonas. And then they walk to another spigot and they see a clay-like material, and that organism is called Gallionella, which thrives in iron-rich water, and it excretes the soluble iron into a insoluble iron. It's an iron oxidizing chemolithotroph, and it excretes ferric hydroxide. And by chemolithotroph, we mean that it sort of uses as its electron donor, it uses, instead of NADH or an organic molecule like we do, it uses an inorganic.
Rocks. It eats rocks.
And most of these bacteria that I'm going to talk to you about are chemolithotrophs. They use as their donor at the top of the electron transport chain bacteria. They use inorganic instead of NADH. So they can, obviously, they need to have carbon metabolism of some kind to make more bacteria, but they get their electron transport and generate ATP by reducing minerals, not inorganics mostly. And basically, Osborne's groups and others have really shown that the Earth is full of microbes that are chemolithotrophs that basically live, use these inorganics and transform them. And by transforming one mineral into another kind of mineral, you can generate all sorts of different things. And these are the things actually we find when we mine Earth's surface or the crust.
And if I could put a sharper point on that, they estimate that the biomass, all these microbes living down in the rocks and chewing the rocks, account for 10 to 20% of the entire biomass on our planet.
Yeah.
So all the people, the animals, the worms, the fish.
Yeah, so it's really amazing.
These microbes, do they go throughout the entire crust or just the top part of it? Do we know?
It's hard to say because we have only gone kind of a tiny way, right, into the depth.
We're only a mile in, Vincent.
How big is the crust?
Right here, they're a mile in. I think the farthest people have gone is maybe 2 miles in. So 2 miles is...
So it's 9 to 12 miles in thickness.
Like 4K, maybe a little under 4 kilometers.
Interesting.
3.2 kilometers. I can't... It's not very... 3.2K. So that's not science, that's running that allows me to do that calculation. So what's really kind of amazing is they not only live down there, which is sort of crazy, but they're transforming it. And they really think it might be possible that the microbes built sort of the ground on which we stand. They kind of transformed things into, very far down, which supports the soil on which we walk, which is kind of mind blowing because again, we think about the atmosphere as being completely transformed by microbes. This is super well accepted. The only debate is which microbes were the first ones to sort of start generating oxygen, as far as I can tell. But here it's this idea, like the ground on which we walk is essentially created by microbes.
The Russians have drilled 7.5 miles into the crust, which is about 12 miles thick. So.
Okay.
Wow.
And how deep do you get, like how far is it from, the ground on which we walk to the center of the earth?
It says on the quick Google, I did a Rich Condit. The quick Google says the crust is thicker and older and can be 20 to 30 miles thick. It's made up of less dense rocks like granite and can be even thicker in such areas as the Tibetan Plateau.
Okay, that's just the crust.
That's just the crust.
Yeah, exactly.
But the problem is it gets warmer as you go
right,
deeper.
It gets much warmer because the center of the earth is very hot. Yeah. So anyways, that's sort of how he starts, is in this elevator. But this article goes on to talk about how we've known about deep dwelling microbes for a long time. In fact, they were first identified in coal samples taken from about 3,600 feet. People discovered viable bacteria in permafrost in 1911. Somebody also about the same time revived bacterial spores trapped in a Pennsylvania mine. And in the 1980s, they knew that bacteria were in groundwater thousands of feet deep. The problem was that, when we go to other planets, we have to people then started sterilizing the equipment they used to go that deep because you could easily introduce microbes from the surface deep. So the sort of good science really started in the 1980s. So Thomas Gold actually speculated that there might be up to microbes 6 miles deep. And that actually raises this idea, right, that is sort of a little mind-blowing as well, which is that if we have microbes that deep, it's possible, possibly even probable, that life might have started that deep and that other planets might also have microbes that deep.
How could they not, really? How could they not? When we read this, how could we assume that, like, our moon or Mars will be sterile?
They need water.
Yeah,
they need water.
They need water. Well, there's definitely evidence that at least there's water underneath on Mars.
Yeah.
I think there's pretty good evidence. So, well, at least our microbes need water. Maybe their microbes don't.
Right.
They need some form of liquid because the chemistry still lay you odds is probably soluble and not solid state.
Right. They need some form of liquid. Probably, yes. So some of the very deep samples that have been taken from 2.8 kilometers down, about 1.7 miles, there's an organism called Desulforudis audaxviator. And that's from a line in a Jules Verne novel, Journey to the Center of the Earth, which is actually pretty cool. And they're described as baguette-shaped. So they're long, rod-shaped bacteria, and they have a single polar flagella, from what I can tell. They live at temperatures of about 60 degrees centigrade, which is about 140 Fahrenheit, so bun-warming temperature, basically. And they acquire energy from radioactively decaying uranium. So again, they have to somehow get carbon, to make more bacteria, but they're, instead of using NADH to generate electron transport and ATP, they're using essentially degradation products from decaying uranium. They fix carbon, so they can use inorganic carbon, carbon dioxide, carbon monoxide. They can also fix nitrogen, although they mostly don't. They seem to have good nitrogen supplies from what they can tell. You think, this is going to be such an exotic organism. It's actually part of the low GC gram-positives. They're in the Clostridial group, which means they're closely related to Bacillus subtilis, which is sort of a gram-positive model system, many lab study. It's actually one of my favorite organisms. And these used to be called the firmicutes, but now have been renamed the Basileota. So again, we talked about the one that was part of the gamma proteose, but again, these guys are part of the firmicute/Basileota. Again, a really large group of diverse bacteria that live in many different niches. They've found microbes in water that hadn't seen the surface in over 1 billion years. This group, Barbara Sherwood-Lawler at Toronto, she really thinks of this work as exploratory, like we're only just beginning to understand what's down there. But it really starting to change how we think about how the planet was shaped. Where did our planet come from? Why does it look the way we do? And beyond just like the soil that we can see, but the whole planet. The article goes on, it talks about Lovelock's Gaia hypothesis a little bit. And this is this older hypothesis, the idea that the Earth itself could be thought of as a living creature that absorbs, stores, and transforms energy. But I think it is important to think like all of these organisms function together, not just in a small ecosystem, like a common garden kind of ecosystem that, a traditional ecologist would study, but the whole Earth is a giant ecosystem. It's just kind of disparately connected. And they talk finally sort of near the end about some scientists who learned rock climbing, sort of rudimentary rock climbing, in order to do some of this exploration, because you're really caving, and many of these kind of research sort of sites are really caves. And one of them actually got an eye infection from some rust-colored fluff that she found, Penelope Boston. But they found these microbes can seem to be actually creating the caves, because the microbes, so these limestone caves, this one was in New Mexico, but they're all around. They're really probably leftover coral reefs from long, long ago. But the microbes eat oil deposits and hydrogen sulfide and produce sulfuric acid that carves away the limestone. So the estimation is 5 to 10% of limestone caves are actually created by these microbes just by their excretion of the sulfuric acid, which carves away the limestone.
It's all about reduced minerals becoming oxidized minerals, and the microbes are using them for food.
Well, they're actually using them not as food in the way that
they're
we use
getting
food.
Their energy.
They're using them for energy. When I teach microbiology, I try to separate those two, because when we learn microbiology, when we learn about the electron transport, it's always connected to glycolysis and the TCA cycle and NADH and NADPH, right? But for microbes, a lot of them, possibly most of them, actually don't worry about NADH and NADPH. They recycle that through fermentation anaerobically by excreting things like lactate or ethanol or things like that, acids. But their electron transport, instead of having NADH, as we've all been taught at the top of electron transport, they might have hydrogen sulfide at the top of electron transport. As long as the acceptor is below the donor on that tower that we learned and then were traumatized by on the electron tower, then they can generate energy that way to make ATP, generating the energy and the reducing equivalent to generate that, right, is actually what's transforming. They can take hydrogen sulfide and make sulfuric acid. So that's really exciting. And so sort of near the end, they talk about how young Earth probably had very low mineral diversity, maybe some quartz or things like this. But now we have 6,000 kinds of different minerals. And they speculate, and have good reason to speculate, that some of our most precious metals like gold and silver are basically created either directly from this reduction reaction to generate ATP, and also to generate proton motive force for transport, but also the release of these waste products like hydrogen sulfide, which then react with the minerals. So it sort of blows my mind. And also this paper mentions a paper that we did back, I think in TWiM 291, I think it was Michael's snippet, about how microbes might contribute to plate tectonics, which at the time was like also a crazy idea. But if you kind of think of it in this context of this larger giant Earth-size ecosystem, you can imagine how the microbes kind of doing this pretty deep in the Earth, deeper than 2 miles or even 6 miles, but farther down, might actually be big contributors to moving big things. It's like tiny local effects add up to big things when you have, I don't even know how many microbes. I can't do that size math.
And also that the microbes are changing the geology, right?
Yeah, it's just amazing.
When I was reading this, it reminded me of the One Health paradigm.
Yes.
That we talk about it, especially like with infectious disease and that we're all connected. And so what you put on your crops, ultimately is going to affect what people are exposed to. But now it's like the one Earth hypothesis, right? Or not hypothesis.
It's happening.
A model. Yeah.
No, exactly. It's a one Earth, right? So our common garden needs to be much bigger if we're really
Can any of these microbes be cultured, Petra?
I think many of them, actually, they have managed to culture them. I think, they've fixed some of them, they've isolated already as essentially monocultures. The D. Daxviator has, I think, was the only microbe they found in there. So I think they can culture some of these. It's not as necessarily, some of the problems with the culturing microbes has been that they grow with other bacteria or archaea, and they need to grow together just for the delta G of their reactions to work. I think here it seems like they've been able to culture them, or at least get enough of them to get very high level sequencing. I
remember a long time ago, Elio did a paper about bacteria that dwell in rocks, and he said the dividing time for some of them was 1 year.
Yeah, I would not be surprised if it's very long. I think some of these, the gamma proteo and the one from the Basileota, the Clostridial relative, I think is probably a little faster than a year. It probably depends what they're eating. I know that some of these very ancient, really rooted archaea grow really long, like the Loki archaeota. But in any case, it's just amazing to kind of think about these bacteria. They've had billions of years to grow and transform the Earth. So.
I wonder if these depths of the crust and below are the one place that humans can't mess up.
Oh, I'm sure we can mess it up. I think that's how do we know? Like when we go to another planet, how do we know we're not accidentally bringing something along with us? It has to be super sterile to know that it's not caused there. It looks like, from what they're saying, there's these formations of microbes that are big enough they can tell. Like if you see this rusty, fuzzy stuff everywhere, you're not so worried about your tiny bit of contamination, at least in terms of identification.
It might take a while before we learn how to culture all these creatures, but whole genome sequencing can generate some just fantastic phylogenetic trees to try to understand what is happening down there. Wow.
I was really amazed by that. So many of the ones, at least he highlighted in this article, are actually related to bacteria we know really well, like groups of bacteria they're not new groups of bacteria in the sense like their bigger families are well understood.
And in principle, the bacteria can be very deep because the ones at the bottom of the ocean live under boiling conditions, right, essentially. So I bet they go down past the crust even. Who knows?
Possibly. You've got, because then you've got pressure and temperature, I guess. Yeah. Although I don't know. Yeah, I don't know. I've always thought of like the pressure when they talk about in the ocean, right? Because you've got pounds, water, all that water on top. So I don't know when it's air what the pressure difference is. I imagine it's much less. But probably if you go 30 miles deep, it's pretty high.
Norm Pace, when he was working on hydrothermal vent organisms, asked us poor graduate students at the time what it would be like to grow at pressures at depth of 15,000 feet, like the Marianas Trench, where he was recovering the microbes. And what Norm pointed out to us, and Norm is a well-trained biophysicist, pointed out that the dielectric constant of water, the solution chemistry, changes at that pressure. And so I don't think the majority of biologists know how to react. So what does it mean when the dielectric constant of water changes? What does it do to the fundamental biological chemistry? I certainly couldn't answer it as a graduate student. I doubt I could take a stab at it even now.
Yeah, I mean, and I don't know, but here we're dealing with air pressure, not water pressure.
Yeah.
Which is going to be very different.
Speaking of the Marianas Trench, Michael, a phage has been discovered 8,900 meters down in the trench.
Another thing, how do they get so deep? They just sink?
Well, they're infecting hosts down there, right?
Yeah.
It evolved down there.
Yeah, but that's like the deepest phage ever found.
Yeah.
Amazing.
It's wild.
Okay.
One Earth.
Thank you, Petra.
Oh, you're welcome.
Now Michael is going to talk about bioelectronics. Bioelectronics.
Yeah, today on TWiM, I'm going to introduce us to what I affectionately refer to as bionic microbes. So, as a child growing up in the 1970s, I was fascinated by the show The $6 Million Man, which premiered in 1973. In the 6-year run of the show, we saw how the main character, who was literally rebuilt with remarkable prosthetic devices that enhanced his physical performance characteristics, subsequent injuries he sustained from a test flight of an experimental aircraft, accomplished remarkable things like saving the world in every episode. Today, I'm going to take us on another fanciful journey, but this one is science fact rather than science fiction, and it offers hope to the initially at least 125 million global sufferers of psoriasis, an autoimmune disease that also afflicts approximately 3% of the adult U.S. population, or about close to 8 million people as of 2020. My poor late mother suffered from this condition, and I can offer that she was willing to try almost anything to relieve her suffering from this disease. So this paper will likely bring hope and wonder and questions from folks suffering from this condition. Now, this paper has 4 complex figures with 70 supplementary figures, and it appeared in the May 31st issue of Science, the one with the cuckoos on the covers. That's how I generally sort my sciences, by the cover art. And so the cuckoos you can sort of find pretty quickly. The title of the paper, as Vincent alluded, is Active Biointegrated Living Electronics for Managing Inflammation, and was authored by a team from the University of Chicago, Columbia University, and Rutgers from the departments of chemistry, molecular engineering, biomedical engineering, and the James Franck Institute at the University of Chicago. There are 9 authors of the manuscript. The first 2, Ju-Young Shah and Se-Hin Kim, equally contributed to the work as first author. If the science paper wanders too far afield from your scientific comfort, there's a terrific commentary by Peter Olfsson entitled Living Electronics Resolve Inflammation, which is pretty much the story that the authors were able to show, and it's pretty much what sufferers from psoriasis want. They want the chronic inflammation that is literally growing skin to stop. Now, the majority of microbiologists will acknowledge that inflammation is a component of our coexistence with microbes that live on us and in us, with inflammation being a key component of our antimicrobial defenses. And wound healing, where our immune system, through an elaborate signaling opera, manages to eradicate pathogens, clear out the cellular debris made by the pathogens, and ultimately restore us to our old selves. Again, this is a dynamic process that is accepting and responding to the ever-changing local environment, where the tissue and organs, as well as the microbes, are all communicating in what some might view as a seemingly undecipherable cacophony of biochemical regulatory signals. Left unchecked, inflammation can lead to a very debilitated host. And in fact, non-resolving inflammation is an important contributor to the development of prevalent diseases that you all know about, like atherosclerotic cardiovascular disease, pick any autoimmune disease, obesity, failure to recover from severe infections, and certain types of cancers. And as our colleague Daniel Griffin often reminds us from his weekly clinical updates, it's the inflammatory phase of COVID that is often responsible for many of its ill effects. In our most simplest understanding of the immune system, it is a complex multi-component network of biochemical interactions that are continuously balancing these complementary yet contrary forces that ultimately result in the concept of homeostasis. Now, during the inflammatory process, sensors for danger, which are located on nearly every cell in our bodies, become activated by something called pathogen molecular patterns, commonly abbreviated as PAMPs. that are in turn are appreciated by these pattern recognition receptors on our cells. Now, the PAMP may be bacterial, viral, fungal, or even parasites. In the case of bacteria, these PAMPs can be things like peptidoglycan, lipoteichoic acid, and for fungi, zymosin, which is a beta-glucan, which is similar to peptidoglycan. So as an example, the authors offered that when the bacteria invade the skin of a mouse, this action then triggers an activation of an inflammatory response, where even the peripheral nerves similarly detecting the bacterial components, and then they are simultaneously signaling to turn off the response. And the activation of select neural circuits that in turn can accelerate the resolution of inflammation, or using the metaphor again of an opera, where everyone singing ultimately promotes the audience enjoyment of the opera, or in the case of our bodies, survival, healing, and that return to homeostasis. So with that as background, what is a bionic bacterium? No, the authors did not build prosthetic flagella or pili into the microbe like the creators of the $6 Million Man did when they were rebuilding them, but rather they used electronics and then off-the-shelf bacteria, literally off-the-shelf, to mimic.
Off the skin, Michael.
Well, off the skin. Off the skin, thank you. To mimic the innate homeostatic regulation that is necessary to down-regulate our immune system. And they were lucky because these off-the-skin bacteria, Staph epidermidis, is what they used to model their system. And then Staph epi, as it's affectionately known as, can actually down-regulate inflammation. So their idea was, let's put Staph epi in a bag and tell it when to grow and stimulate and turn off the inflammatory signal. Now, the way they approached this is they used a mouse model that would address psoriasis, where it's nothing but chronic inflammation that is responsible for the condition. And in the psoriasis model, they're simply going to measure the thickening, or through the electronics, the electrical conductance. As the skin gets taller, growing on top of itself, it doesn't conduct electricity as well as normal skin. So what they did is they developed this active biointegrated living electronic system, which is a fancy way of saying, as I already jumped to, they encapsulated the Staph epi into a hydrogel matrix that in turn was incorporated with an electronic array to combine the treatment and continuous monitoring of the psoriasis disease progression into this single device, which they then principally used to measure the disease tissue through impedance, humidity, which is also a measure, because I remember my poor mother would remark on how her skin was just sloughing off. It was like chronic dry skin was just literally falling off. So they're measuring the humidity as well as the temperature. Now, some diabetic sufferers out there already have integrated electronics embedded in their skin that continuously monitors their interstitial glucose concentration. And then that electronic device communicates with a continuous insulin infusion pump that acknowledges the glucose concentration perceived by the sensor, and then using onboard fuzzy logic algorithms, they dispense the proper concentration of insulin diabetics need to safely manage their glucose concentration. And I'll bet you this is what the authors hope to achieve with their device, or as Peter Olofsson offered in his title, Living Electronics Resolve Inflammation. Now,
but can I just say, like, how audacious this idea is? And I want to credit the authors for being bold.
Oh, this is bold.
Staph epi lives on our skin, and most of the time it's not a problem. But it does cost the U.S. medical system over $2 billion annually when it gets into the wrong place at the wrong time and flares up and can cause
disease.
Yeah. So it's really bold to say that we can basically harness this and encapsulate it in this device where we can control it and not have it escape into the tissue.
Well, that's a very good point, Michele. The key to their design was the use of a hydrogel, where this water and energy-filled matrix can serve as an intermediate layer, serving as a bridge, if you will, between the mechanical gap between the electronics, which is effectively on top, and the biological system, which is based in the bottom, which they showed then provided the necessarily cellular communication function for the tissue modulation or modification, which is illustrated in Figure 1, panels B and C. Now, biological systems such as bacteria and mammalian cells naturally exhibit this cell signal generation and transmission, which has been leveraged by the bacteria for many years to get the host to do things. That's, after all, what quorum sensing is all about. Now, the platform they developed, they named ABLE, which is an active acronym for Active Biointegrated Living Electronics. Again, they selected Staph epi because it's well tolerated on our skin, while at the same time, it being able to provide to the bioelectronics the capabilities to get information it can use to regulate inflammation and promote skin regeneration. All of this is dependent upon an empiric observation that Staph epi is good for skin generation. Now, I dropped a few references into the show notes about Staph epi and its role to control inflammation. Now, they built their system so that they will feed the Staph epi, and they know that the Staph epi modulates the skin immune environment, while the bioelectronics will coordinate the electrical signaling, collecting information from the skin, and then manage, this is what Michele was getting at, manage the biosafety of the bacteria. And again, check Figure 1B to see what they do. And it's got a lightning bolt to show you that they're going to effectively use energy or charge to prevent Staph epi from transitioning from a commensal to when it's no longer a commensal and it becomes a pathogen. So the gel matrix serves to prolong bacterial storage so they can pull it off of a shelf, slap it on your cirrhotic lesion. The bacteria continue to grow because they're feeding the suckers. And to some extent, the matrix's viscoelastic properties ensure stable skin interaction. And the gel is composed of gelatin and tapioca starch. And that's what Figure 2 is all about. The experiments where they tried other materials, and that's beautifully illustrated. Bottom lining this, they now have bacteria in a gel matrix that are stable. And in future, you can imagine how you would unseal one, place it on this flare of psoriasis to encourage your skin repair. Or you may even think about using this in burn victims where they have significant inflammation. So how did they test, if you will, their bionic bacteria for their ability to alleviate a flare of psoriasis? Well, again, this is beautifully illustrated in Figure 3. Again, it's a complex figure with 9 panels, alpha through India, where you'll learn that using a mouse model of psoriasis, they were able to address whether the therapeutic bacteria in the form of the Staph epi, through the presence of its lipoteichoic acid and the toll-like 2 receptors on our cells. And my immunology colleagues will often serve to remind you, toll-2 is the gatekeeper. It controls whether or not we're going to have full-blown inflammation. Now, the experiment they did was straightforward once they had the device built. They applied the ABLE device to the cirrhotic skin for 4 days and learned that the signal-to-noise ratio from recordings of a 6-lead EKG, or electrocardiogram, from the mouse was largely enhanced from day 0 to day 4. So it effectively is reducing conductance that they are seeing. They offered that such a diminution in signal in the surface ECG suggested an improvement that the living components of the patch contained within the ABLE system played an important signaling role in mitigating the symptoms of psoriasis. Now, they're doing a bioassay, folks. They're measuring psoriasis. And this is seen beautifully in figure 3, panel H. And importantly, the impedance of the psoriasis skin lesion, again, you don't have to be a pathologist to see the graph is simply going down. And when it goes down, it indicates recovery of the psoriasis. And as my mother often offered that her dermatologist told her in the mid-90s, it's simply uncontrolled skin growth. That's all psoriasis is. So lower impedance suggests less skin or a return to normal. These results also aligned with the psoriasis severity index, which dermatologists use to measure the severity of disease, and they did this in 5 independent animals. So they next created a battery-free, wireless, printable, flexible circuit board that can achieve this comprehensive interplay. If you will, they are building the equivalent of an insulin pump, but for the control of psoriasis. The device principally works by monitoring skin impedance, humidity, and temperature. And then for safety, on-demand bacterial disinfection through the application of current that in turn likely leads to an uncoupling of the membrane potential of the microbes, which in turn leads to internal free radical generation, of course, bacterial death. They also, and this is what all the microbiologists and immunologists want to know, they also address the issue of mechanism. How do the bionic bacteria, if you will, do this? Or how does ABLE modulate the cellular environment during the inflammatory skin conditions through their biogenic cues provided by the SAF-EPI? Well, this takes us to figure 4, another multi-panel wonder, but surprisingly, a very easy to walk yourself through. In panel A, you have the control, no ABLE, and then you have the ABLE skin samples, where you can easily see in a simple H&E stain how ABLE was able to substantially reduce the pathological damage of seborrheic skin lesions in the ABLE-treated mice. When you then get into the immunohistochemistry analysis, this panel 4B, you clearly see the notable decrease in macrophage activity after ABLE treatment. They continue with this figure to drive home that ABLE device is doing its job and that CD31 immunofluorescence and immunohistocompatibility staining both again revealed fewer dilated blood vessels, which you need to make more skin, in ABLE-treated samples after 4 days compared to the control mice. Similarly, they throw in some cytokine analysis profiling showing reduced levels of, wait for it, the inflammatory cytokines, with notable reductions in interleukin-17, interferon gamma, and tumor necrosis factor. And every introductory immunology student knows IL-17, interferon gamma, and TNF are bad actors of the immune system, especially when you throw in IL-1 on top.
Well, you want them at the start.
You want them at the start, but you don't want them prolonged.
Just shut it down once they've done
I had
their
a question. I'm looking at this in figure 3. I guess it's figure 3G, where they have the electrode and everything. So, I mean, this is really amazing. But then the question is, how practical is this? Like, how big is this thing? And if we're looking at, I guess, skin on, it looks like somebody's wrist here. In 3G, is the little circle the implant? I mean, how?
It is. They're going to have to go a long way to optimize it. It's not the insulin pump that our friends at Dexcom and some of these other insulin pump makers are getting, but it's the theory that they're getting to. They're beginning to dissect the immunological signaling where they're using bacteria signals to effectively turn down the regulation. And the last bit of coolness, Petra, is that ABLE modulation is needed to modulate the skin microbiota, because it goes back to sort of your paper. What are the other bacteria doing in this complex community, bringing home the one earth or one health complex? And the modulated skin microbiota induce transition from a cirrhotic state to a healthier state. And again, they give us what a microbiologist wants. They give us species level, where they observed a low abundance of staph epi on the skin, which is in panel 4F, indicating the low staph epi related safety concerns under the bioelectronic control. And furthermore, the abundance of Staph aureus, the species that most cirrhotic patients get infected with and becomes a problem. When you infect your cirrhotic lesion, it's often evil Staph aureus that's doing the bad deed. They also notice abundance of other skin microbes such as Rhodococcus and Delphia, which were also modulated by the ABLE device, concluding that altogether, changes in bacterial abundance can probably be attributed to this mutualism between different bacterial species and the staph epi. And they conclude figure 4 with an expected mechanism model, where in panel J, they put it all together for us, where they describe that ABLE can coordinate the regulation of the inflammatory skin environment. And that really gets to Petra's observation of how practical is this. I think once we begin to figure out how we can miniaturize and control inflammation, we may be able to go from the skin to other areas where chronic inflammation is significant. And
sure. And of course, they chose the skin because it's so accessible. So as an experimental model, it's perfect. But then they can take what they learn and target it toward particular diseases or conditions where it's much more confined, like IBD or something like that.
So, Michele, did they, I know you wrote to the authors, did they share how they came up with this idea?
Yeah, thank you for bringing it up. So the two first authors, I'll start with Jun Shi, was a graduate student doing this work who got their bachelor's in science and chemistry in China and then came to University of Chicago to do their PhD. But he describes that what really inspired them is that there is a literature of using helminths, so microbial worms, let's call them, to treat ulcerative colitis. So that's an autoimmune disease with a lot of inflammation. And there is data showing that you can introduce parasites or parasite eggs into the intestine and it dampens the inflammation.
Yeah, it's amazing.
So that was the inspiration. And then, of course, by using an animal model, the mouse model, they could have it be much more accessible.
No, that's really exciting because there is all this, like, especially with helminths and other things modulating the immune system. I would not have made that connection to this paper.
It's
but
PAMPs.
Really
it's PAMPs, folks.
I know.
So he pointed out, the now Dr. Shi, who's recently started his postdoc at Stanford, but he pointed out it is audacious to think that we can take a opportunistic pathogen and actually use it as a therapeutic. But he said it's precisely these challenges that make the journey of scientific discovery so thrilling. And the pursuit of innovative solutions, even when they seem crazy, drives progress and fuels his passion for interdisciplinary research. So he grew up in an academic environment, pursued chemistry, but has always been interested in more kind of interdisciplinary research. And he really feels fortunate that he was able to join the lab of Boji Tian, his supervisor, who offered unwavering support, insightful guidance, and encouragement that helped them through the toughest times in the research. And he aspires to one day continue his own independent career and eventually lead his own lab, focusing on innovative solutions to complex healthcare problems. And one of the people that he worked closely with was another graduate student, Se-Hyun Kim, who earned their bachelor's in chemistry at the National University of Singapore, and also did a summer research experience at the University of Cambridge with the Reisner Group, studying microbial biohybrid systems for solar fuel synthesis. So again, really futuristic application of microbes. He's now a third-year graduate student investigating this interface between artificial materials and microorganisms and developing therapeutic applications. And in his free time, he enjoys, he's an avid fingerstyle guitarist, and a street musician, and a YouTube music creator. He also enjoys watching films, exercising, cooking, and traveling. I also wanted to highlight that some of the funding they got for this really creative work comes from the Bill and Melinda Gates Foundation. They got a grant titled Electrical Control of Vegetative Bacillus subtilis and Spore Dormancy, and also from the Department of Defense, the U.S. Army, Living Bioelectronics for Electrifying Microbial Mammalian Communities and the Skin, Gut, Brain Axis. So it's wonderful to know there are funders out there that will give this really creative, audacious work a chance to come to life.
Yeah, I dropped into the show notes a paper that was in Science Robotics in April of 23 called Bio-Inspired Ingestible Electroceutical Capsules for Hunger Regulating Hormone Modulation.
Ooh.
And so this is not as wacky as it seems. Bottom line in all of this, these authors created something as cool as a $6 million man, for they have demonstrated how we can dragoon commensals, put them in a baggie, feed them a little, and then they can promote therapeutic effects through a living interface. The key, as Petra brought up, is going to be miniaturization and the potential for creating a systemic effect.
And they also added a safety shutoff valve, right?
Oh, yeah.
They have it designed so that they could
electrocute them.
Yep. They added a particular agent that would trigger the pulse and just sterilize the whole unit, keep it safe.
Amazing. It's just amazing.
It is amazing. Microbes, not only do they create everything, we can use them to do everything.
Apparently, yes. Just a matter of...
this is going to get the crazies all crazy because now we're really putting chips with bacteria.
I know. It is a literal
Well, if
chip.
you can come up with a chip to control appetite, there's a big
There's a big market for that.
Which, of course, is also a serious health issue. So.
Oh, yeah.
For sure. Thank you, Michael.
You're welcome.
That is TWiM 314. You can find the show notes at microbe.tv/TWiM. If you enjoy our conversations, we'd love to have your financial support. Go over to microbe.tv/contribute. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin's at Washington University. Thank you, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.